Okay, we'll start now. So today we will learn one of the web tools which is open source, freely available, anyone can use, and that is called as Kegg, Kegg. And when we search Kegg in Google, we will get this result. Click on the first result, www.genome.jp. And when you click on this, you will get this interface. Now, the thing is, this Kegg, it was designed by some Japanese uh, organization, and the thing is it is collection of databases. It is not a single database. For example, as it says, uh, under Kegg databases you have a list. If you can see the screen, there is Kegg pathway, there is Kegg bright, Kegg module, Kegg orthology, genes, genome, virus, organisms, compound, glycan reaction, enzyme network, disease, drug. So there are a lot of databases, and also there are some analysis tools from Kegg Mapper to SimComp. So it's a collection, but the one which is quite famous, I would say, is this Kegg pathway. And this Kegg pathway, it is a collection of different pathways. For example, I think BBT people, you are going to learn biochemistry next sem. Yeah, I think we have learned these things in 12th. Glycolysis and what was that? Krebs cycle. Yeah. So all those different pathways. So in the same way, it is not just limited to, metabolism of carbohydrates, proteins, or lipids. There are other pathways as well. For example, it might be either metabolism related pathway, whatever the chemical that we take, or as a drug, or whatever we consume as food, how it gets metabolized. There should be a pathway, right? From compound A, it will be degraded in a structural flow to get the final desired product. And the second one is it might also contain genetic information processing. For example, pathways that are involved in transcription or anything related to the gene processing part, and also related to cellular processes, organismal systems, and also human diseases. For example, there might be a disease which affects the particular system in our body, and it affects a particular pathway. And also drug development, it is a collection of different pathways which fall under these seven broad categories. And again, there are some keywords. The thing is, every pathway or every entry in this database is going to have a unique identification number, and this will help you uh, in identifying. If there is just identification number there, by just looking at that you can just say, oh, this might be you know a map. For example, here it says map is manually drawn reference pathway identifier. For example, I'll just show you when we look into that. If you just scroll down, so the first section is metabolism, and under metabolism you have few pathways. One is global and overview maps, and there is subdivision. It starts with carbohydrate metabolism and then energy metabolism. These some of the pathways, for example, glycolysis, gluconeogenesis, citrate cycle, pentose phosphate pathway. All these things you will learn in biochemistry. And again, you have nucleotide metabolism, pyrene and purine and pyrimidine metabolism. And then there is amino acid metabolism, and there is vitamin and cofactor metabolism, secondary metabolites, etc. And also, as I said, genetic information processing. Under this, you have transcription related pathways, translation related pathways, and folding, sorting, and degradation. This folding, it refers to protein folding, how primary structure it transforms into secondary structure, then tertiary structure, and finally quaternary structure. And then replication and repair. Have you guys completed genetics? Have you? I think no. So in genetics, you will learn in detail about different types of repair mechanisms. Yeah, you will learn all these things from non-homologous, homologous, mismatched nucleotide-based DNA replication. So don't worry about these things much. And then under environmental information processing, it focuses on signal transduction. For example, if there are two cells, if one cell produces a chemical and that chemical travels and it attaches to the receptor on the other cell, after that, how does the signals get transmitted? Am I making sense? So all these things, hedgehog signaling pathway. I think if there is someone from MSI, there is a course called as developmental biology, and I vividly remember this hedgehog signaling pathway. It is there in developmental biology. I think it also came in the exam. And you have different signaling pathways. And then coming to cellular processes, how does catabolism happen? Can anyone let me know what is catabolism in the chat? Today's session is going to be pretty short. It's not going to be lengthy. Okay, correct. Now, under cellular processes, you have pathways related to transport, catabolism, cell growth, and death. Again, I think if you have completed cell biology, you might have learned cell division in detail. And also, I think you will learn little bit about signal transduction as well. And there is cell motility related pathways. These are very important concepts, bacterial chemotaxis. And then under organismal systems, as you are well aware of, there are different uh, systems in our human body. There is digestive system, there is circulatory system, nervous system, immune system, endocrine system, etc. So again, pathways related to those particular systems are listed here. And if you scroll down again, it has listed few famous diseases here. There is cancer, cancer related pathways, and specific types of cancer: colorectal cancer, pancreatic cancer, specific pathway related to pancreatic cancer. If you just click on this, for example, you will be redirected to this page, and whatever the diagrammatic representation that you can see, it's in the form of, it looks like a flow chart. This is the pancreatic cancer pathway. Are you guys able to understand? Okay. So the thing is, one homework for you guys is, you can see, if this is a pathway, there are few cells which are in green color. And within that green colored boxes, rectangular boxes, if I can zoom in, the font is black, but font color is black. And then there is one rectangular cell here that you can see where the font is in red. So what does this change in the color mean? That is a homework for you guys. And again, so here the arrows will indicate the direction of pathway or the reaction, like what was the initial compound and what it formed. And it also lists enzymes. Here, I think there are no enzymes listed. Okay, I'll just show you when there is an enzyme. And again, these big representations, for example, P13K AKT signaling pathway. This just tells you that there is one more pathway called as P13K AKT signaling pathway, which is connected to this pathway, but it is not directly related to maybe pancreatic ductal cell or this pancreatic cancer. So it is just mentioned in this box. Are you guys able to understand? Whatever is displayed here, this is the main flowchart, and whatever is not listed, which is written in the form of a box, MAPK signaling pathway. These are some related pathways. And it is also here, you can see apoptosis is there. And then there is ERBB signaling pathway. It is not that even I'm not understanding everything written here, but I'm just trying to explain you what are all the things that are present here in the result. So if we can go back, and there are few other diseases: immune disease, neurodegenerative disease, Parkinson, Alzheimer. So even I think we have learned in previous sessions that whatever is going to be stored within the database is going to be manually curated. Like there are scientists who will study, who will clearly evaluate and approve it before it gets deposited in the database and displayed here. And the thing is, if I click on Alzheimer's disease, whatever that you're going to see, this is just what we know as of now. Are you guys able to understand? As the research continues, things get updated, and this Kegg database get updated. So you have even skeleton based classification, etc. So let's just look at one simple example. Let's go for glycolysis. I think this is something all of us will understand. Am I right? So if you click on glycolysis, you will get this. Everyone got the same flowchart? Okay. So whatever you can see here, this is the flowchart, and you can see there is starch and sucrose metabolism here, and it gets converted to alpha D glucose 1 phosphate, and then it gets further converted to alpha D glucose 6 phosphate, and then D fructose 6 phosphate, D fructose 1,6 biphosphate. Are you guys able to understand this? I think this is what we have learned in 12th. Yeah, okay. So you have the whole pathway here. So this is the simple one. And again, as you can see, you have pentose phosphate pathway here. This just means that this is connected to the glycolysis pathway, but the main focus here is on the glycolysis path. Even there are citrate cycle mentioned here. There is propanoate metabolism. So these are some related pathways that are connected to this main glycolysis pathway. If you click on this, you will get the pentose phosphate pathway, hopefully. Yeah, you can see. So it's a related pathway. It is not the main pathway that we were looking for. Now, as you can see, whatever the substrate or the chemical compounds, they are named here. It is clearly written alpha D glucose 1P. If you can see my cursor, then just left to that, there is a circular dot. And if you click on that, you can see the structure of alpha D glucose 1P. And whatever you can see, I hope we learnt these type of representation of chemical structures in 12th. Am I right? I think this dark black, it says something, and this dashed line says something else. I think it's more related to in the 3D space whether it is present front or it is present backwards, something like that. Yeah. And here you can see a number 5.422, 5.4.2.2. So this is nothing but enzyme identification number. If you click on that, you will be redirected to a page which will be part of Kegg enzyme database. Do you guys remember? I mentioned Kegg enzyme also. It is one of the databases. So it will be redirected to that. If you click on this circle, which is next to alpha D glucose, you will be redirected to Kegg compounds database where you have information related to alpha D glucose. Now, on my left, you have an option of scaling. So if I just decrease this, the size of the flowchart decreases. If I increase this, you have, the original size of the flowchart back again. So in the search here, you can search for the compounds that you want to uh, maybe see. For example, if I want to check uh, arbutin, here you can see on the left arbutin. If I want to check if it is present in the pathway, I can just write arbutin and I can click on go, and it says there are two arbutin words present in the flowchart, and it also highlights where exactly it present. Can you guys see? It is highlighted in pink for me. Arbutin extracellular and arbutin 6 phosphate. Are you guys able to understand? Okay. Now, here under modules, here what happens is this is, the whole pathway, and this whole pathway is divided into different divisions. So if I click on carbohydrate metabolism, you can see a particular pathway within this whole diagram will be highlighted. So this represents the carbohydrate metabolism. But as there are different subdivisions here, if I want to know gluconeogenesis, if I just select this module, this will be highlighted again. So whatever is highlighted, that is representing the process of gluconeogenesis. Are you guys able to understand what is a module? Module is like dividing the pathway into subdivisions, and you store it as glycolysis Embden-Meyerhof pathway and glycolysis core module involving 3 carbon compounds. So whatever is highlighted here, it is involving only 3 carbon compounds because I have selected that part of the glycolysis pathway which involves only 3 carbon compounds. Hope you guys understood. Shall I go ahead? And what exactly is a network? We will look into this later. And then there is one more thing I can show. There is show description option. If you click on that, you have a description for glycolysis. It says glycolysis is the process of converting glucose into pyruvate, and this whole paragraph, this is present. And if I click on organism group, it just gives you, as glucose is going to be consumed by all different kind of organisms, there might be some slight difference in the way in which this glycolysis works, or there might not be. So here it is just a list of uh, glycolysis pathways in different organisms. Here you have for mammals there are 170 entries, of which primates alone contain 32 entries. And yeah, there is bird related glycolysis pathways, there is reptile related glycolysis pathway, crocodilia, and there is amphibian related, fishes related, etc. Are you guys able to understand? And again, if you just change the group here, you get a different set. In group 1, you have this 753 entry for animals. If you click on group 2, you have one subdivision here. If you click on group 3, one more subdivision appears, so that you can clearly understand. Under animals, there is mammals, and within mammals, there are these many listed. Hope you guys understood. Let me just go back. Now, if I click on, let's just click on this D fructose, or maybe this one, the first one, alpha D glucose. If I click on this circle, I'll get this, and this is nothing but Kegg compound database result. Here I have the ID for alpha, for this D glucose 1 phosphate. The Kegg compound ID is C00103. And again, you have different names for the same compound listed. They can be synonyms, or it can also contain some common names. For example, this coriester, it's a common name, I guess. Whereas this alpha D glucose 1 phosphate, this is the scientific name. And you have the chemical formula for the same, and you have exact mass of this compound, and you have molecular weight. So what is the difference here? So this molecular weight is going to be the average of the isotopes of the compound. Are you guys aware of what are the isotopes? Yeah, okay. So you just take the average of molecular weight of different isotopes of the same compound, and that is going to be the molecular weight. And again, you have structure here. This is the chemical structure. And sometimes, do you guys remember in Swiss ADME, there was an option to draw the structure of the chemical compound? Am I correct? Yeah. So there might be few bioinformatic tools where you can input chemical molecular structures, or the files that store the structural information of the chemical molecules. So you can download the structure in two different formats. One is MOL file format and one is KCF file format. So you can look into that as well. And here you have all the reaction IDs. There is a separate database called as Kegg reactions, and every reaction gets an ID. And here it has listed all the IDs where, or all the reaction IDs where this molecule also is a part of. Are you guys able to understand? In all these listed reactions, this D-glucose 1 phosphate plays role. It is present in one of the positions in the whole path or the reaction. And again, you have pathway here. So there are these many pathways where this D-glucose 1 phosphate has been identified or noted. And you also have the pathway names. And I think I need not explain this again. What are modules? And again, the enzymes that act on this compound. So you have it has listed all the enzymes, and this is unique enzyme ID. And you have network. I will look into this later. Yeah, we have other databases. This is just cross-referencing for the same molecule. You can click on these, as these are clickable, these are links. You can go to those databases and you can look into these compounds. So this is all about Kegg compound. Are you guys able to understand? Okay. Now let's go back. Now let's click on one of the enzymes. So let's just take which is right after alpha D glucose 1P, 5.4.2.2. If I click on this, I'll again get a result, and this is what I have got. Have you guys got the same? Have you guys got orthology? Okay, so what is orthology? I think this is also something you will learn in biochemistry, but I'll just explain this in short. So, what happened is during the course of evolution, let's say there was, what can I give as an example? Anyone knows what was the name of the organism from which monkey evolved or monkey came from? Okay, let's just forget that. So, during the course of evolution, what happened was there were few genes, and as a particular organism evolved into different species, and eventually over the period of time they even separated completely, and they started belonging to different genus and families. What happened was these genes, although they got transmitted, their function was same. Am I making sense? For example, if there was a hemoglobin gene, that gene got transmitted to the you know the next species that got evolved, and but the function of the hemoglobin remained same. What is the function of hemoglobin? It is related to oxygen delivery to cells, right? So the hemoglobin present in chimpanzee is also going to deliver oxygen, and hemoglobin present in humans is also going to deliver oxygen to cells. Are you guys able to understand what is ortholog? Yeah, these set of genes which get birth from the same gene, but they are present in different species but perform the same function. Are you guys able to understand? I hope I'm not complicating things. So, like, they originate from the same gene and they get transmitted to different species, but their function remains the same. And these set of genes, they're called as orthologs. And you have some information related to orthology here. And again, you have modules listed, you have different pathways for the same, and you also have disease. So in glycogen storage disease, this enzyme also plays a role, or this enzyme is also present in that pathway. And even in congenital disorders of glycosylation type 1 and muscle glycogen storage disease. So in all these diseases, this phosphoglucomutase plays role, or it is present, and its representation ID is 5.4.2.2, but the chemical name is phosphoglucomutase, and it has a symbol as well, that is PGM, and it has an ID for orthology, that is K01835. Hope you are able to understand. And what is bright, this we will look into it later. There is a separate database called as Kegg bright. And again, genes. So as I said, these are different genes and they belong to different species as well. But the thing is, their function is related to the function of phosphoglucomutase. Or these are different genes present in different organisms. They code for phosphoglucomutase. There might be like slight difference, but what they do is same. They take part in this glycolysis, and they do the same role or same job as phosphoglucomutase. That's why you can see even the short representation for the gene, it is same, PGM, PGM, PGM everywhere. Are you guys able to understand? Okay, now if you just scroll down, you can see there are a lot of genes listed. Okay, so again, you have different orthology related information. I think you have more than two, three orthology related data. If you, in the end, you have enzyme related results. Hope you guys got this one. Enzyme 5.4.22. Okay, yeah, so you again have the entry and it says it is an enzyme and the name of the enzymes. And again, class, even enzymes are classified based on, their functions or based on their roles. And you again have reaction related informations. Even it specifies that it belongs to Kegg reaction database. And again, the substrate related information on which compound it acts and what it forms, the product related information. And then there is a comment related to the enzyme, and it says maximum activity is only obtained in the presence of alpha D glucose 1,6 biphosphate, and this is an intermediate in the reaction being formed by a transfer of phosphate residue from the enzyme. You can read this. This is some information related to the enzyme again. And then you have some enzyme history. This is nothing but uh, today we have, let's say, 10,000 enzymes that we know. In future, because of scientific research, scientists identify more enzymes. Let's say they identified 10,000 more. So totally there are 20,000 enzymes. So in that case, the 20,000 will again be classified based on whatever they have. They might create a new enzyme class or they might merge two enzyme classes. So in that case, the identification number changes. Hence, it says enzyme 5.4.22. It was created in 1961 as this ID that it has given 2.7.5.1. But in 1984, after almost 20 years, it was transferred to this ID. Are you guys able to understand? Okay. Now again, you have pathway related information in what are all the pathways this enzyme plays a major role. And again, orthology related, which I explained, and gene related, genes that code for this enzyme phosphoglucomutase. And again, some scientific references. As I have always mentioned, in all these databases, whatever information is present, that is going to be based on the scientific research. So you have some IDs that if you click on this, you will be, I think, redirected to some journal articles, as it says, reference author's title journal. And then you even have some external database cross referencings. And you also get one more result related to reaction. And this we will look into it later when we learn Kegg reaction database. Did you guys understand this Kegg enzyme and Kegg compound and Kegg pathway? Even the orthology thingy we will look in detail in the future sessions. I thought of only covering three today: Kegg pathway, Kegg compound, and Kegg enzyme. Are you guys able to understand? Okay. Now let me just show you one more way. So whatever we did so far, we always, like, we chose the compound from the pathway, we chose the enzyme from the pathway. So we used this pathway result as the initial source. We don't have to do that. Even if we go here, there is something called as Keck compound, and it says it stores small molecules. if you click on Kegg compounds, you can search the compound by the C number. The C number is nothing but there was an entry number, right? C00 something. That is the C number. And even you can search the name of the compound by name, or you can even search the name of the compound by formula. And even you can restrict the results based on the mass. Are you guys able to understand? I think I'll just leave this as homework so that I think this is easy. You can just type the name, you will get the result. And again, coming back to Kegg, what was that? Kegg enzyme. Yeah, here you can write the name of the enzyme and you will get the result again. For example, I'll just copy paste this. This is one of the enzymes. I'll copy paste and you can see there is result. I have got two result and I'll click on the first one, and if I click on this I'll get this result. As we went through pathway, we got ortholog, reaction, etc., etc. But if we go through the Kegg enzyme database directly, we'll not get much. We'll just get this output. If you want to see the reaction, you can click on this reaction cross referencing, etc. Are you guys able to understand? Okay. And one more. For pathway, you can just type the name of the disease as well. If I just write diabetes, you will just get all the pathways related to diabetes. Here it says this will redirect you to type 2 diabetes mellitus pathway, and the second one will redirect you to type 1 diabetes mellitus pathway, and this third one to maturity onset diabetes of the young. Do you guys remember? I think we got the same result when we were going through Uniprot disease and variant section for insulin. I think we got same type 1 diabetes mellitus, type 2, and this maturity onset diabetes of the young. Yeah, so there are multiple databases, they just link every database together. And again, you can search pathway even through organism names. For example, if I want to know something related to E. coli, maybe I did not write the name correctly. Wait, just go to Kegg pathway. Yeah, there is organism, and if I write the organism here, yeah, I think this is the scientific name. There are multiple strains of E. coli, I guess. I'll just select one and I'll just write select. Wait. Okay, I got. So these are different pathways. Wait. Okay, so I just wrote cholera, and we have got Vibrio cholerae infection, biofilm formation Vibrio cholerae, related pathways. You can even search by name of the disease or even name of the organism. For example, let me just write Vibrio cholerae here and I search, and again you have got a set of pathways where Vibrio cholerae organism is a part of. Are you guys able to understand? Okay. So this was all, guys, for today's session. I think we can finish the session early. Hope you guys learned something in today's session. And yeah, any doubts? Okay, hope there is no doubt. Good night, have a great day. See you tomorrow. Hope your FFCS went well. Okay, glad to know. Bye bye.